Whenever you're ready.
Hi everyone, so today I'm going to talk about the genus Amansia. So I'm just going to start with their classification. So Amansia is in the kingdom Plantae, class or phylum Rhodophyta, class Florideophyceae, order Serramiales, family Rhodomeliaceae, and genus Amansia. So there are 44 species within the genus overall, and then there are 3 that are found in Hawaii. Some general characteristics of the genus include having a rosette of serrated blades. The main axis is corticated, and they're also uniaxial. And then going into their reproduction, they are procarpic, and they exhibit isomorphic alternation of generations. So in terms of their life history, they have sporic meiosis. I couldn't find a diagram that actually depicted Amansia on it, so this is a different genus, but overall it's basically the same thing, but they start out with a dioecious gametophyte. They don't have separate sexes, but then they release their gametes. They have their carposporophyte is procarpic, so they grow their carpospores, release them, and then they have a tetrasporophyte that looks similar to their gametophytes. Then the tetrasporophyte releases spores and turns back into the gametophyte. So there have been a couple of interesting studies done on the genus Amansia. This is one of the ones that I found. It's a pretty complex study, so bear with me. But basically they found that Amansia contains lectins, which are proteins that can block nutrient absorption by binding to sugars. So they found that lectins can possibly block pain or nerve stimuli, and so the word for that, or the more complicated word for that, is antinoception. So they tested the effectiveness of lectins on blocking pain or nerve stimuli on Swiss mice. So they did 5 different tests on them. I'm not going to go into detail on the tests. They really weren't very nice tests, but I thought overall it was like a really interesting study. So basically overall they found out that these lectins were really, really effective in blocking pain and nerve stimuli. So in the first test, they basically induced pain in the mice, and actually prior to starting any of the tests, they dosed the mice with lectins, and then they waited about 30 to 50 minutes. So it's kind of like if you took Tylenol and then you waited 30 minutes or something. So they dosed the mice with lectins, and then they did these series of tests. So in the first one, they found that when they did induce pain, the lectins inhibited the pain by 80 to 85%, depending on how the lectins were given to the mice. So they were either given orally or they were injected. In the second test, they injected formalin into one of the feet, and then they looked at how the mouse reacted in terms of licking, because that's like a pain response to like lick their wound. And it was actually more effective than morphine, given at a lower dose, so that's pretty impressive. They also did a thermal stimuli test, so they gave the mice lectins, introduced them to a heat source, and then they tried to see how long it would take the mouse to respond, and they found the mice responded much more slowly, so they basically didn't realize that they were being introduced to pain, so it kind of blocked that nerve response. And then they also did a sleep study with the mice and found that the lectins increased their sleep duration. And another really interesting part was if they were given the lectins and then they were secondarily given naloxone, which blocks opioid receptors, they actually did experience the pain still. So there's some sort of interference with the opioid system. So that's something new that they found out about that. And then just quickly, I'll go over this one. They another study conducted on the same species, Amansia multifida. They found that this genus has like a really high antioxidant content, and they were able to inhibit beta carotene oxidation by 40%. And so since these algae also have low cytotoxicity, they might be able to use Amansia multifida as a source of antioxidants for humans. And that's everything. Does anyone have any questions?